So I'm going to be talking today about methodologies to try to understand the genetic architecture of complex traits. And here, by complex traits, I mean any type of trait that you can imagine that it's common in the population, it's probably complex. And the reason why we call it complex is because there are a bunch of genetic determinants together with a bunch of environmental factors that all work together to either increase or decrease the risk for disease. And today, I'm going to be, because we're interested in the genetics, I'm going to be interested mostly or entirely on the genetic basis of complex traits. So I'm going to be interested in the genetic inherited factor for disease risk. Now, for the past 10 years, a lot of people have been interested in delving into these questions. And the tool of choice has been in terms of genome-wide association studies, which essentially looks at the genetics of a bunch of people that have the trait and a bunch of people that don't have it, or disease and controls, and tries to find places in the genome that could explain that difference, as highlighted somewhere in this significant association. And without further ado, these types of GWASs have essentially been conducted for any common disease that you can imagine and have produced thousands, if not tens of thousands of points in this map. So probably you have seen this image before, which is showing you the fact that GWAS has been conducted for a given trait and has been found a region in the genome, for example here, that contains some variants that increase or decrease risk for that particular trait. Okay? So obviously, we can continue to do that further. We can. You probably will notice that this is, now it's 5 years old. So the latest image here, it's probably too hard to show for you. So obviously, we can continue to do that, and we can continue to pad this image with more and more dots. But today, what I'm going to be talking to you about is methodologies that go beyond just finding significant associations. So methodologies that go beyond just mapping regions in the genome that may harbor disease variants for various diseases, towards trying to understand more about what is so special about particular regions in the genome that increase or decrease our risk for disease. Okay? And I structured this talk mainly comprising 3 different topics here. And I'm trying to go through them quite on a fast note, with the idea that this is more of an invitation for you guys that are in the room to help us if you're interested in these types of methodologies. Particularly, I'm going to mention it. These work from students in my group, and they're somewhere in this room, and I'm going to mention it across the talk. And please feel free to come to us. We feel like we've made some significant improvements to the methodology, but we feel like there's a lot of other work to be done. So please, if you're interested, we're very, very eager to collaborate. Okay, so first, I'm going to be discussing methodologies that we've been developing for the past couple of years of how to add functional data. And here, I mean literally adding annotation data that comes from large projects like the ENCODE or the roadmap project to try to disentangle disease risk. And then in the second part of the talk, I'm going to talk about how do we go about not just finding single nucleotide polymorphisms that are associated to disease, but actually finding the susceptibility or the target genes as a follow-up in post-GWAS studies. And finally, I'm going to talk a little bit about new methodologies that we're very excited about that try to look at how can we put together variants or how can we put together signal across many variants, try to understand what's so special about those. Okay, so the first part of my talk, I'm going to be talking about ways where we can put together these types of data, which is functional partition of the human genome as created by the large consortia as roadmap or ENCODE, that essentially for the purpose of my first part of the talk, you can think about it as simply marking the human genome based on plausible functionality with respect to some intermediate phenotype. So for example, you might think about one such functional annotation of the human genome would be where the genes are. Or you might think about regulatory regions in a particular tissue. And there's a lot of ways that you can detect that through various protocols, which I'm not going to go through them. But the idea here is that now, 2 years or 3 years after the roadmap or the ENCODE project have been completed, or they're mostly completed, we have for every region or every variant that you can imagine in the human genome, we have thousands and thousands of putative functional scores that this project has generated us. So now in the past couple of years, we've jumped from not really having good functional annotations, but towards having an overabundance of functional annotations, to the extent that you can randomly select any region in the genome and you'll find a lot of these functional annotations. So our main interest here were with respect to common disease, which of these functional annotations are actually relevant for whatever disease that you're interested in, right? And as a good approximation towards that, you can start counting where the GWAS hits, where they lie, or you can start going towards what we like to do in terms of heritability-based methods, okay? And this is what I'm going to be describing throughout in the next couple of slides. So heritability makes a natural sense here, because by definition is just how much genetics, or the proportion of variance in the phenotype that comes from the genetics. And here you can dissect this further and further, but here I just want to make the point that there are two ways of defining the narrow sense heritability. One is the total proportion that comes from the entire genome, or if you are interested in just a particular set of markers, you can refine that definition and define a different quantity, which is the SNP heritability, which is how much of variance explained comes from a specified given set of markers in the genome, in this case SNPs, right? So note that these two quantities are different, and more so after we sequence everyone, this quantity will get closer and closer so the SNP heritability will get closer and closer to the total narrow sense heritability. But by definition, because the number of SNPs that we're looking at is much smaller than the total variation in the genome, this number is bound to be smaller than the total narrow sense heritability. Okay? So how do we go about estimating these? Well, we go through mixed models, and there's a lot of ways to explain mixed models. The simplest way that I like to think about them is that now, once we measure genetic variation in multiple sets of individuals, and we have their phenotype, now we can contrast the sharing at the level of the genetics with the sharing at the level of the phenotype. So we can essentially look at the genetic variation in this room, and we can ask whether people that tend to be more similar in their genetics, whether their phenotype, whatever phenotype is also more similar or dissimilar, right? And mathematically, this goes through this, we take the genetic variation, we build a genetic relationship matrix, which encodes this amount of sharing between pairs of individuals, and then we essentially fit a multivariate normal distribution, which tells us how much is this matrix essentially explaining in the phenotype. Here, the phenotype is a vector of phenotypes across many, many individuals, right? And the idea is if this quantity is large, then genetics explains a lot. If this quantity is 0, then genetics doesn't really contribute much, right? And you see why this makes a natural sense if we think about a functional partition in the genome, because now we can ask, by looking at pairs of individuals, we can ask whether they're more similar or dissimilar at specified locations in the genome, right? So we can ask, for example, if these two sets of individuals tend to be more similar than two random people in the population at coding regions versus enhancer regions, right? And then we can play the exact same game as before, and now instead of just one matrix of encoding the relationship between pairs of individuals, now we have 3 in this case, or we can have as many as we want, right? Which is, this would be the sharing between pairs of individuals restricted to coding regions. This will be to the enhancers, and so on and so forth, right? And now we can throw them all into a variance component model, and we can let them compete for variance, with the idea being here that if most of the causal variance, or most of the variation that contributes to the trait, tends to co-localize with these red regions in the genome, then this quantity here, this sigma squared red, tends to be high, while these other quantities tend to be close to 0, right? That's kind of like the intuition behind this types of modeling. And we've done this for lots of traits and lots of data. Here I'm showing you some recent work where we looked at 60,000 people with, or 6,000 men, half of them with prostate cancer, half of them without, and we were trying to find regions of the genome that contribute the most to the heritability of prostate cancer. And here we looked at 600 or so functional annotations in the genome coming from the ENCODE project. Here I categorize them by DNAs, acetylation, methylation, and so on and so forth. And you can see that what I'm plotting here is the expectation. So this would be if there's no enrichment for signal in these regions in the genome, you'd expect most of these dots to be by the diagonal. But every now and then you see one of these regions or one of these functional elements that has an overrepresentation of variance in this variance components model. And this will be an indication that that the causal variants tend to colocalize in regions that are marked by, in this case, by DNAs in LNCAP, which is a prostate cancer cell line. So that makes a lot of sense biologically, right? Same thing with the acetylation. And we can take this, so this is one interesting way of finding what functional annotations make sense for the disease of interest, but we can take it one step further. We can start partitioning the total variance into different subgroups in the genome, right? So now, if you can imagine, we have the human genome, and we start partitioning it into a hierarchical cluster, hierarchical structure, where first we assign variation to the coding or coding proximal regions, which is like a couple of percent in the genome. Then, you know, the acetylation mark in prostate cancer lines, then the androgen receptor binding sites, and then, you know, DNase hypersensitivity sites in all the other cancer lines. And then we can leave the rest, 84%, as maybe not that interesting for prostate cancer in this case. And then this is kind of like a visual representation of this Pac-Man shaped like partition of the human genome, right? Now we can put all these things together into one of those variance components model, and we can try to see, okay, what happens at the level of variance explained in prostate cancer risk. And if we do that, then what we see is that the vast amount of the genome, in this case, so this is like 84%, that they don't really contribute much to explaining differences in inherited risk for prostate cancer, while most of the signal comes from this acetylation mark in LNCAP, which essentially says that most of the common, so this is all based on the common variation, most of the common risk colocalize with enhancers in prostate cancer lines, right? Because this is a marker for enhancers, with an additional signal coming from DNase hypersensitivity sites across most other cancer cell lines. And this was done in 50,000 Europeans in this is the consortium name. We also looked at it in 10,000 individuals of African American ancestry, and this is very interesting because now this is a totally different ancestry group, but we see the kind of a similar functional partitioning in the human genome, kind of suggesting, or being consistent at least, with the fact that the contribution of common variants to prostate cancer risk is actually shared across different ancestries group. Furthermore, it's actually shared in this particular sites or this particular regions in the genome. Interestingly, we also looked at other phenotypes because we wanted to see how much is actually specific to prostate cancer or not. And so here, this is an average across 11 phenotypes from the WTCC consortium. And this is mostly autoimmune traits. And what we see here is that there's a massive decrease in variance there. So this serves as a control, if you will. So we see that variance marked by this acetylation mark really are driving prostate cancer, but not are driving other phenotypes, while DNase hypersensitivity seems to be marking general variation that contributes to multiple phenotypes. So now this gives us nice insights into, in this case, into the prostate cancer. Where should we look next, or what are regions in the genome or functional marks that are useful to investigate? But another way to think about it is that this gives us this natural prior to try to reinterpret the findings that come from GWAS. And if you're familiar with the problem, there's, after you find a locus in the genome, there's a lot of variation there that's correlated with that, and the causal variant tends to not to be the top SNP in the region. So then essentially what you're left out is, let's say here we're looking at 3 regions in the genome for a given phenotype. You see this association statistics come from GWAS. You have this LD structure. There's like putative causal variants somewhere here that are unknown, and this are the things that we want to rediscover. And we can use this functional annotation essentially as priors towards upweighting certain SNPs as being putative causal, right? So the idea here is that by looking at all the GWAS risk variants for a given phenotype, we can actually borrow information across different loci, right? So for example, here we might be fairly confident that this is probably likely to be causal, while here there's an abundance of SNPs that are all equally likely more or less to be causal. So then if we observe this variant to be in an enhancer, then essentially we can borrow that information over here to upweight slightly the enhancer variance there, right? So how do we do this? Well, I'm not going to go through the details of it. We had like several methodologies along this line, but essentially it's a hierarchical model where we can put these functional annotations through a logistic prior or this type of prior that give us the probability of causality for every variant, and then we observe the association statistics conditional on that. So the parameter of interest here would be the effect of every annotation on the causality, the correlation structure at every locus in the genome, and then the effect size of a causal variant. So we extended that type of ideas towards looking at multiple ethnic groups. This is one of the most powerful way that people have done fine mapping studies, and the idea being there that if you look at two different populations, then the correlation structure at every locus in the genome tend to be varied, to be population specific. For example, this is what happens in Yoruba at this locus. So you see shorter blocks here of correlations or LD versus Europeans and Asians. And by putting all these things together, we can further zoom in on the true causal variant here, as opposed to just things that are tagging the causal variant. How do we go about doing that? Well, there's a lot of like a methodology there, which I'm not going to go through the details of it, but essentially if you condition on a given set of causal variants that are the same across all ethnic groups, then you can actually write down models, right, where this data was actually independent of this other data, conditional on knowing what are the causal variants. And then things become simpler. There's like an equation there. I'm happy to talk more about it, but for the time being, it works. It's more accurate than what other methods. This is in simulations. We also looked in real data. You see, this is here we're looking at the number of variants that you would have to restrict yourself to before you're 90% confident that you capture the causal variants. And you see that without annotation in transgenic settings, you more or less need 24 variants to functional validate to find most of the causal variants at a given random locus in the genome, while if you throw in these functional annotations, you'd reduce this to 21 variants, right? And now a natural question is, okay, this doesn't seem like it's a massive decrease, right? So it's like you're jumping from 24 to 21. There's not a huge increase in accuracy by looking at all these functionality. Why do we bother, right? And one of the ways to think about this, or that I like to think about it, is that this quantity here, it's kind of aggregating a lot of things, but it's not really the best metric to look at. For example, here's what we did this analysis in transethnic rheumatoid arthritis, and these are the top variants that we found in this analysis to have posterior probability over 0.8 to be causal. Okay, so let me highlight some of these variants. For example, you might find variants that if you look at in single population analyses, they don't really have a good posterior probability to be causal, so you won't really pick them up. But when you look in transgenetic, the posterior probability jumps really, really high. So they're consistent across multiple ethnic groups. That's one thing that you might find. Another way to think about it is if you see, for example, this variant happens to fall right within a functional category that was deemed very important for rheumatoid arthritis, and its posterior probability jumped from 0.2 to 0.9. So this is variants, individual variants that by themselves you wouldn't have really picked them up if you were just analyzing the data without functional categories. But now the functional data it's putting a huge priors to overcome the association signal. So these are ways where we can integrate functional annotation data that comes from ENCODE and roadmap towards prioritization or towards finding causal variants for different complex traits. But ultimately, these analyses will actually give us we'll zoom in on single variants or single mutations in the genome, but won't give us a causal hypothesis, right? And from that perspective, it might be more interesting to think about what if we take our unit or biological unit from a single mutation in the genome towards a causal gene, okay? And in the second part of the talk, I'm going to describe some of the methodologies where we try to move away from just looking at one SNP at a time or one single variant at a time towards looking at the entire gene, okay? And to think about this, in GWAS, what we typically do, we associate hundreds of thousands of individuals, or we associate variation in hundreds of thousands of individuals with disease status. And if we find a significant association, we call those as risk loci in the genome. Now, if you want to do the same things but looking at gene expression, well, you have a problem, right? So these sample sizes are usually much, much smaller than in GWAS. So if you associate genetic variation with the gene expression, that's usually referred to as EQTLs, but you can also associate gene expression to disease status directly. The issue being there is that people don't really do that because these sample sizes are orders of magnitude smaller than GWAS sample sizes. If you think about the entire GTEx project, which is a massive undertaking, only has maybe 1,000 individuals, while any complex trait that you can imagine, probably GWASs are over in the order of hundreds of thousands of individuals. So there's a massive discrepancy between the sample sizes, which essentially says that small weak effects between these things you won't have power to detect them. Okay? And when we were thinking about these types of problems, this fits directly into a missing data problem, right? Because now I have this type of data. Now all that I need to do is just fill in this component of the data, right? So you can imagine coming up with just a statistical model or a computational model where you take this type of data, you build a predictor from genetics to expression, and then you take that predictor and you start filling in all these quantities for every individual. So you impute the expression of every gene, and then after you've done that, now you can, so you do gene expression imputation. After that, you just take this data and do the genetic association. So now you do association between transcriptome or between gene expression and disease status, right? Or what we like to call transcriptome-wide association status. Now, obviously, there's several other groups thought about related things too. And why we thought about this from two perspectives. One is that you might actually find new regions in the genome, but more importantly, you might be able to do a better job of fine mapping or zooming in at a given locus on the right gene of interest. Okay? And what exactly are we doing here? What exactly are we testing when we're correlating the imputed gene expression with trait? Well, we're rejecting this type of null where there's no real connection between this gene expression and trait, and there are lots of ways that you can think about it. And then essentially, if we reject that null, then we're left with at least these alternatives, right? Where you have this genetics impacting expression impacting trait. This is really what we're aiming for. But you can have the other ways too, where you can have a SNP that impacts a trait, which impacts gene expression, or you can have so-called pleiotropic, where you have a SNP or a genetic variant that impacts both gene expression and trait in the same time. Okay, so anytime you do association on the imputed gene expression, essentially you're left under these categories. And you need to do further follow-up to figure out whether you're in this case or the others. For this particular work, we did a lot of simulations, and in most of the cases, we think this was the case, but this needs to be, it's kind of worry about these types of prediction. Okay, so how does it work? Here's the cis expression or the genetic variation locally around a given gene. This is the expression. You just come up with whatever genetic predictor or whatever predictor that you want to. Here we chose linear genetic predictors, or blobs, if you will, for very very specific reason. But nevertheless, you can pick whatever type of predictor that you want that connects these variables with expression. And then now that you have genetic variation for different individuals that you don't have expression, but you have trait, okay, from GWAS, then you just apply your genetic predictor one at a time, and then you just correlate the imputed expression with the trait, okay? And this is, again, this is work that we've been doing, as well as people in the GTEx consortium and other people as well. Okay, so the first question is, that sounds really, really interesting, but does it really work? And the short answer is that yes, it works really, really well. And the reason is because the genetic effects locally on expression are much larger than GWAS, so you need much smaller sample sizes to train that model. And furthermore, if you just restrict yourself to like the top genetic variant that is associated to expression, that usually, on the average, explains roughly half of the entire variation. So what this is saying is that if you just take the top SNP that associated to expression and just use that as a proxy for gene expression, then you only lose roughly half of all the power here, right? Which might not be that much, given that now you jumped from 1,000 individuals when you have people with expression in EQTL studies to hundreds of thousands of individuals when you look at the expression. Okay, so then it works. This is we looked at both different technologies. We looked at RNA sequencing data in 600 individuals as well as microarrays. We looked at different tissues. This is blood and this is adipose, all together in like 3,000 more or less data set. Okay, so it works. We think it works really well. But really the novelty in our mind was that because we're using a linear predictor, now you can expand the same machinery from linear genetic predictor without really needing every individual's genome or every individual's genetic variation data. And the really cool part about it is that any linear combination of genetic variants is actually translating really, really well to a linear combination of effect sizes from SNPs on the trait of interest. So what you end up doing is you train a linear predictor from an EQTL type of data, and then you just take the marginal effect sizes from GWAS. Okay, so you take like the p-values, for instance, from all the SNPs in that region, and then you apply a transformation, a linear transformation, after you account for the correlations between these variants, and then you get what would be, or you can impute directly the standardized effect between that expression and trait. Okay, so you don't really need to go one step above and every individual impute expression, but you can just impute directly the correlation between expression and trait had you imputed expression to all the individuals. Okay, and that sounds like it's a mouthful. Does it work? Well, we had data with 1,000 individuals where we had all these three measurements. So this is the young Finn study where we had blood expression as well as height as a phenotype. And then we did both ways in parallel. And then we compared one versus the other. And this is what you get. So this is, here I'm plotting you the standardized effect size between expression, the association of expression and trait. If you compute it based on imputation one individual at a time versus imputing it directly from p-values. So, and you see you get a correlation which is essentially 1. So there's really not a lot of there's not a benefit from looking at individual level data. So where does this type of idea work? Well, it works in general when you have essentially, if you compare a GWAS versus just testing the top EQTL for a given for any gene versus this new imputation-based transcriptome-wide association scan, or TWAS we call it, you see that so this is the genome-wide association sample size in tens of thousands. This is the power. You see that first you gain because now you're testing far fewer hypotheses. So you're not testing 1 million SNPs. You're just testing, 10,000 genes. So this is the gain in power. But if you have a single causal variant that impacts expression, then essentially it's more or less the same thing as just taking the top SNP that impacts expression. But if you have multiple SNPs that impact expression, which in turn impacts trait, then you have a massive gain in power by aggregating the effect size of multiple SNPs through the variable of gene expression. Okay. Now, obviously, if that's not truly the causal mechanism, so if genetic variants don't impact trait through expression, you're not going to catch it. So this is not a replacement for GWAS, but this is a different analysis that one could do with genome-wide association data. Okay. So at that point in time, we're still very skeptical. So what we did, we started reanalyzing old GWAS data and we found new things in that old GWAS data, and then we looked at newer GWAS data to see if those things would have been found, and in short, yes. This is in lipids. So essentially, we found 25 genes in the 2010 lipid GWAS with 100,000, which were found subsequently when they doubled the sample size. So essentially, we are getting more and more convinced that all these things are finding robust association, and it's not just finding false positives. So where are we here? Well, Nick Mancuso in my group, who's over there somewhere, so you can talk to him, he started essentially analyzing lots of traits with all the data that we have available. So here we looked at essentially all the GTEx data set, which is like 40 tissue plus another 4 or 5 tissues with large sample sizes, in lots and lots of complex traits. And here's a bunch of them. This is, you can see the GWAS sample size are anywhere hundreds of thousands of individuals. And this is like a short summary of all the new genes that were found. And then kind of like the way these numbers break down is the following. If you look at only the GWAS reported loci, here in this data set we had like 1,500. Most of them would actually have a gene that's significantly associated with some genetic variant. So that's what the E gene is. So there's a gene that is genetically controlled by some genetic variants. But most of those genes are not really associated to traits. Most of those genes just happen to be at these loci. So another way to put it is co-localization between eQTLs and GWAS will give you some ideas about the genes, but most of those have nothing to do with trait. So you really need, if you jump to like associating those genes with trait, only maybe like 1/3 of them would actually have a association to disease risk, right? What does that mean? There's a power issue here involved. It could be that this gap is explained by the genes having a smaller effect on trait, but most of it, I think, or we think, it's simply because all the genes in the genome will be E genes. So all the genes in the genome will be genetically determined. So this overlap will be 100%, but obviously not all the genes will contribute to that disease of interest, right? So only a fraction of those. And then, going one step further, in 200 or so cases, we are zooming in on exactly one TWAS gene at every locus. So this will be great hypothesis in our mind for testing, because now we are finding a single gene that fits a lot of criteria. So it's both has an eQTL, it's also associated to disease risk, and this will be a great place to start, okay? So obviously now you can just keep on doing these things and you can tally up all these numbers, but that might not be that interesting if your goal is not to just come up with all these gene lists. Another thing that someone could do is try to correlate genetic or complex traits through the gene expression. So the idea here being is how much of the genetic relationship between different traits is not coming from random regions in the genome, but it is due to their imputed gene expression. So the goal would be how much of sharing at the genetic level between, I don't know, BMI and HDL, for instance, is not due to environment, but is due, and it's not due to the overall genetics, but it's due to just the expression part of or just due to the gene expression, right? So this is what Nick has done, and this is what I'm plotting here. This is like all these 30 traits by all these 30 traits, and obviously all of them are correlated with them at level of 1. And you see these nice clusters that make a lot of sense. So for example, these are our blood phenotypes. These were these are our blood phenotypes too. You see this Crohn's disease, IBD. So there's a lot of things that make a ton of sense here. And furthermore, actually, if you repeat this analysis, but you run a method like LD score, which ignores altogether expression, essentially you get roughly the same, you get pretty much the same result. So you get, here I'm plotting every dot here is a pair of traits. And with some difference in significance, you see that you can recapitulate all the genetic sharing or genetic correlation between pair of traits that LD score uses without any idea about the expression through just looking at the gene expression, right? And there's a lot of like suggestions here. There's a lot of like fuzziness, and we're trying to understand this. But this is consistent with most of the genetic sharing between complex trait being driven by the local cis genetic control of gene expression. And this is what we're going forward with this. But probably the coolest thing that you can do with this imputed gene expression is try to use them as instrumental variables in trying to understand whether there's a causality statement here, right? And I'm plotting here, every dot here is a gene, and here I'm plotting the effect size on triglycerides versus total cholesterol, right? And you can see why I'm choosing these, because total cholesterol is triglyceride plus something else. So there's a simple causality that we know here, right? So we know that this guy here causes this, right? And there's a lot of details that I'm missing out here, but let's say that we're trying to distinguish between these two models, where you have a lot of genes that impact triglycerides, you have a lot of genes that impact the total cholesterol, but then you have a causality, either this way or that way. And let's say that we're trying to compare these two things. Well, obviously, if I just look at genes that impact this guy, okay, and if that thing is true, then genes that impact this guy will have the same effect on TC, right? So there'll be a strong correlation in their effect size. And that's not going to be the case here, okay? So if you condition on one side, then you can see the effect. So if you condition only on genes that impact TG, then you should see a correlation in their effect size with their effect on TC. If this is the case, but you won't see it in this case, okay? So let's see, does it work? Well, this is what happens if you condition on genes that are significant for TC. So you see that this line is decreased and it's overlapping 0, so it's not significant essentially. But if you do it the other way around, you see that this is actually the effect size, or the slope of this is actually increased, right? So it's slightly higher. Of course, there's confidence intervals there, right? So this is consistent, or this could be used essentially to predict that triglycerides are causally impacting TC, right? Which is obviously, that's not really interesting. We knew that up front, but can we apply it to other things? And the short story is that yes, when we're diving through these things, Nick is trying to investigate all these. There's a lot of questions here about multiple testing and so on and so forth, but we do see evidence of like BMI causal impact in triglycerides, or triglyceride impacting LDL, and a bunch of others, which we're trying to understand further. All right, so in the final part of the talk, I'm going to shift a little bit gears and try to move towards aggregating this type of signal, not through the lens of gene expression, like I was showing you before, but just in an unbiased fashion, okay? So how can we take multiple variants and then just add them up together without having the gene expression as weights there, okay? So how do we go about doing that? Well, you can do that through mixed models, but as I was making the point before, large-scale genetic data is not really available at the individual level data, but it's typically available at the level of p-values, or in other words, of summary statistics. So essentially, GWAS consortia usually post the effect sizes in their standard error, but they don't give you individual level data, okay? So how can we group, or how can we figure out the variance in or the proportion of variants that's contributed in trait that comes from multiple variants in the genome, only from summary statistics, okay? And you can start playing with the math, and here you can do two things. You can assume that the effect size are randomly distributed variables, which is the standard mixed model ideas, and the whole, there's a huge literature there. Or you can start treating them as fixed effects, and this is something that we think it makes way more sense than treating them as random effects. So let me recast this. When we go about a given disease, it's defined by its effect sizes on trait, which are supposed to be fixed. They're not going to be randomly generated for every individual that you go sample in the population. What's going to be the random variable here? It's the individuals that are coming from the population. So if you make that assumption that the effect sizes on the trait, which are here beta, are fixed, and the random variables are the genotypes, or the multivariate genotypes for given sets of individuals, then you can easily decompose the variance in the phenotype as this quantity here, where the variance in X is nothing more than essentially the LD, or the covariance between pairs of SNPs, right? So you get this very easy quantity here, which is another way to define SNP heritability, as I was noting before. And obviously, if you have this, then who cares? You can get this. You can quantify this. Now, the really interesting part here is that we have access to GWAS data, okay? And in GWAS data, when you run, a regression between every SNP in the genome and on phenotype, what you get a quantity, which is the effect size from the GWAS, which will, under infinite sample size, will not really converge to the true causal effect. And this is really something that's fundamental for, the whole LD score and everything that we do here. So if you run infinite sample sizes in GWAS, you get an effect size for a given variant that's not truly the causal effect, but it's an aggregation of the effects at nearby SNPs. So this is what this is saying. So the effect size from GWAS is an LD aware, so this is the LD matrix times the true effect size vector, right? And if you take this quantity and you plug that back in, if you have infinite sample size in GWAS, then the SNP heritability, an estimator for SNP heritability, making no assumptions on the effect size on SNPs, they're fixed in this derivation, is this one, right? Now, once you realize, if you're aware of these formulas, you can instantly see that this is a biased type of formula. This is a quadratic form. So this every time there's noise in this estimate, it will add a bias term. But you can easily account for that, and Huembo can like derive all this estimator, which comes from essentially quadratic form theory. You get this unbiased estimator for the variance in trait or the SNP heritability without making any assumptions on the effect sizes. Okay, so this is one locus in the genome. You can extend it to many loci in the genome. I'm not going to get you into lots of details here. Happy to talk more about it, but essentially what you can get out of this is now you can take GWAS data. So and here we took schizophrenia, height, and HDL, and now you can essentially plot them into a scale that's similar across all these diseases, right? So this scale here is how much of the variance in trait is explained by that particular locus in the genome, right? So you have 22 or whatever, 17, 1800 independent or quasi-independent loci in the genome, and now you can do this plotting, and you can actually compare different traits really, really easily, okay? So then, because you have an estimator at every locus in the genome, now you can do other interesting things. Now you can correlate how much variance comes from every chromosome with the chromosome size, right? So this is a natural sign of polygenicity, if you will. This is coming back to the 2010 paper from Peter Vischer's group, where they were showing exactly the same type of plots, where you would have, this is like the small chromosomes 22 all the way to chromosome 1 and 2, and you see that there's a nice trend here. So there's the longer the genome, the more likely that so the longer the chromosome, the more likely it has causal variance for height, and therefore the longer that they explain more of the variance in height, right? You can do the same thing for other traits, and we have a lot of traits that we looked at. Here's a trait where, like rheumatoid arthritis, where chromosome 6 essentially explains a huge quantity and variance in rheumatoid arthritis, which is exactly what you'd expect. That's where HLA is. But you see the rest of the chromosomes still follow this correlation, right? Okay, so we did this across the 30 traits, and you get this type of picture. You get across all the traits that we studied, there's a strong correlation between chromosomal length and variance in trait, or fraction of estimated of the total heritability. And you see these outliers, which are, these are things that are interesting for, or that have HLA explains a lot, right? A lot of variance, like RA, you have, this is like red blood counts, and so on and so forth. You have like traits like the locus of the ApoE, LDL and triglycerides, and you get a complete picture, or much more robust picture, essentially of what regions in the genome contribute to these traits. You can also start aggregating the signal across multiple loci, and you can ask, if I would have to select a given set of loci to explain the most of the SNP heritability, how would that look like? And this is essentially, if you have few loci that explain a lot, then few genes contribute to that trait. If you have lots of regions in the genome contributing a lot, then that's a sign that you have lots of risk loci for that trait. So here's RA and HDL and triglycerides. So half or a fraction of the genome, so it's like 0.2%, explains 15-20% of all the SNP heritability. While here you have schizophrenia and college attainment, where essentially you need 10-20% of all the loci in the genome before you start explaining half of the total SNP heritability. The more off-the-shelf type of equations here, which is something that Huembo in my group has been, she has been developing, you can extend this framework to start looking at multiple traits at once. So you can expand the exact same framework by looking at two traits. So here is like, let's say we're interested in, for example, triglycerides and total cholesterol. You can expand the same framework and you can write down the math, and then you get an estimator that is very, very similar to what we had before, except that now I have effect sizes from GWAS for trait 1 and effect sizes from GWAS from trait 2. And then really the most interesting quantity here is the fact that you need the LD to decouple this induced effect size versus the true causal effect size. So why do we want to do this? What's so interesting about it? Here's a plot that Huembo generated where here's total cholesterol, here's triglycerides. Here, this is the plot of variance in trait explained at different regions in the genome. So that's equivalent in a way with a Manhattan plot, but you don't have this. The p-values are so essentially the variance in trait is a more meaningful metric than the p-values, and you see that. Just by eye, you see that there's a strong correlation between them, but you can actually quantify it. And you see this is the genetic correlation between triglycerides and total cholesterol and triglycerides that comes from this locus in the genome. And just by eyeballing this, you can already tell that whenever there's a locus in the genome that increases or that contributes to both traits, they both go in the same direction, right? So that's automatically telling you that there's a causality that's happening here, right? Obviously, I didn't give you any model here, but there's a statement here by just looking at all these peaks going in the same direction that there's a causal statement that goes through that all the effect sizes tend to be going into the same direction, okay? Now, another thing that you can do here, which is we think it's very, very cool, is you can use this as a way to run to use data from one trait to find interesting regions in a different trait. So for example, here, you run this test and then you find non-loci that have local genetic covariance, but if you look at genome-wide genetic correlation, it's not significant. So you have traits like, for example, HDL and LDL. If you run a genome-wide metric like LD score, it gives you an effect size that's close to 0. So it is, the truth is that the true effect size should be non-zero, but you don't have the standard to detect it. But if you look at it locally, then you can actually find regions that actually contribute to both traits, which is what you would expect. All right, so I think I'm going to stop here. And there's a bunch of conclusions here, but I really wanted to emphasize that most of this work has been done by Gleb Kichaev on the functional integration, by Huembo and by Nick Mancuso, who are actually here in the room. So you can bug them if you want over the week or over the month. There's a lot of open questions here that we need help if you guys are interested in this. So feel free to question us. Any questions?